Okay, so we'll go ahead and get started. We have 3 lectures left, and basically the trajectory of this is today we'll learn one more thing about sequencing amino acids. Then we'll learn the chemical methods of making amino acids, how they're coupled together. And as you saw, there is a metric ton of slides for today. I doubt we're going to get through it all, but basically once you have the sequence together, what happens after that? So primary, secondary, tertiary, quaternary structures of amino acids to proteins that make up enzymes, and then the last 2 lectures will focus really on looking at DNA. So we'll see how everything shakes out in terms of the timing for this, but ultimately where we're going is with your sequence of amino acids, you're going to make another bracelet that will then figure out the triplet codons that encode for those, and then so that'll be an activity. And then since the codons allow for, what's the word I'm looking for? I can't think of it. So an amino acid can have a couple different codons. So you'll come up with 3 different sequences, and then from one of those, you'll make your bracelet. The 5th activity I'm still looking into, and hopefully I'll figure that out over Thanksgiving. So we had looked at cyanogen bromide, which was specific for methionine cleavage, trypsin, chymotrypsin, and carboxypeptidase to generate fragments to ultimately figure out what is the sequence of amino acids in a peptide chain. So one other thing we have to worry about for primary sequence is the presence of cysteine residues. And so those have this thiol side chain. So the SH is a thiol. And what's unique to those is essentially they can oxidize to form these disulfide bridges. And so again, that's unique to SH. Alcohols won't do that under sort of normal conditions. But if you form this bridge, it's called an intrastrand bridge. And you can imagine if you have a sequence with 2 cysteines in it, they form a bridge, that's going to form a really big ring. And so then if you cleave it with some other method, the chain is still going to be bonded by those 2 sulfur molecules, and then you might not get the information you need to predict the sequence accurately. So again, you can get that ring forming, and this is an example of a short amino acid sequence that's generating a hormone called vasopressin. And so that regulates tonicity, which is the difference in water potential between a cell membrane. So as you can see, the N terminus, again, which we were saying is on the left, the C terminus on the right, this NH2 hanging off here, that's telling you that instead of having the acid as the normal, it's basically the amide. So the NH2 means that, but that's still the C terminus. And so within that we have this large ring that again is due to the disulfide linkage between the 2 cysteines. So cysteine will form those that sulfur-sulfur sigma bond with the presence of oxygen and air pretty readily. But again, OH hydroxyl groups do not do that readily. So the chemical method of cleaving that uses peroxyformic acid. So the structure is shown on the arrow. And what happens is the sulfur atoms, the SS bond is cleaved, and then sulfur is oxidized. And so now you end up with cysteic acid. So that's SO3H, cysteic acid. So if, back in the 70s, people would go to the barber or the stylist and get permanents called perms. So basically what they're doing is they're putting chemicals in there that are sort of breaking these bonds to get the hair straight. Then they put it in the curlers, and they're putting more chemicals back in to reform those bonds so that now the hair gets the coil to it. So that's what a perm means. I got my hair cut, and I was inquiring about prices of things to the stylist, and so hair coloring is one of the most expensive things, and so when they do that, they're putting like bleach or hydrogen peroxide to get the color out, and so it's a chemical process to get color out, to straighten, that sort of thing. But again, it's down to a chemical thing why they're putting that stuff on the hair to begin with. So a couple questions here. How many rings does vasopressin contain? So we already know there's one giant ring, but then looking at the amino acids present, how many other rings are there? So we have one there. So we'll say +1. Tyrosine has a ring, that's another one. Phenylalanine has a ring, it's another one. And proline has a ring. So I think overall, this is going to get us 4 total rings. So really the two that really matter are the large one and then the one for proline, because proline is the only amino acid that's a secondary amine. And as we'll see hopefully later today, how that will affect the secondary structure of the peptide. So if vasopressin were first treated with peroxyformic acid, so that's going to cleave this. And then the enzyme trypsin, which goes after arg and lysine, the basic residues, how many fragments would be produced? And again, the way we want to interpret this is start from the N-terminus and then read. So anytime we hit large or lys, we include it and then cleave. So there's an arg, so we would cleave there, and that should get us 2 fragments. So, as we wind up this semester, I'm going to use that that term read, like you're reading it, but also the enzyme trypsin is also reading it in a sense, because it's specific for those amino acids. So as that sequence goes through the enzyme, it's sensing or reading, and it says, okay, here's an arg, now I'm going to cleave. So those, if vasopressin was first treated with peroxyformic acid, we cleave there. And then chymotrypsin, so that senses the aromatic containing amino acids, what fragments would be produced. So really what you're looking for to see is the phi, trip, or tier as a new C residue. So right away we can eliminate A. That was the prior answer. So I think we can eliminate C as well, right? Because that's just having cysteine. So between B and D, notice that amino acids 2 and 3 are both aromatic. So it should cleave when we're reading. It should cleave right there. And then as it's reading again, it should cleave right there. So that would eliminate B to give D as the correct answer. And then it'll just have this as the rest of the fragment. So in summarizing, you know, the concept of sequencing or figuring out the primary sequence, the first thing that you'd actually do is treat with the peroxyformic acid to cleave any cysteine linkages. And then the enzymatic methods that we learned, it says cleave at the carboxy end, just meaning that what they're specific for will end up as the new C-terminal residue. So trypsin goes after lysine and arginine. Chymotrypsin, the amino acid residues, phenylalanine, tryptophan, and tyrosine. And then carboxypeptidase always goes from the right to the left. But as we said, you know, you clip that first one off, that generates the N-1 length, and then that one reacts. So it clips off the new C residue. And then as you'll see in today's quiz, there's a plot where over time, the thing you see initially the most, that's like the C residue of the entire length. And then the new one's gonna grow in over time, but it's not gonna be there as much because you're not starting with it. N-terminal residue were the chemical methods using the Edman or Sanger analysis that put a tag on of known molecular weight. Then you use mass spectrometry comparing to a library to figure out just matching those amino acids. And then the least specific of everything was just digestion under HCl and refluxing water. So really the end one, you know, that can tell you composition. Like, okay, there's these types of amino acids in there in this proportion. But it doesn't give you any information about how they were bonded together. Any questions? So we'll briefly run through some methods of making amino acids in the lab. If you were working in a lab, you would just buy amino acids unless you were doing some non-proteinogenic amino acid that you wanted to incorporate. Then you'd have to use one of these. So for example, sometimes people want to include cysteine instead of sulfur. So this would be a method to make something like that. And all of the reactions are based on things we've seen in the past couple modules or from Organic 1. So the first one is reductive amination. And that's where you're starting with an alpha keto acid, where the alpha carbon to the acid is a ketone at oxidation state 2. When you condense with ammonia, that makes an imine. And then you can hydrogenate that with H2 to reduce the imine. So the strategy here is that you have to make your starting amino acid. This part is basically your side chain. So that has to be incorporated from the very start. So whatever you want that side chain to be, that's what you have to make into your alpha keto acid. So as a note here on the bottom, so you could do this reduction in an asymmetric fashion, where instead of getting the racemic product, you could selectively get R or S. So this would be a way to make unnatural amino acids. One of the reactions we learned from the prior module was alpha bromination. So if you take a carboxylic acid, again, what you'll note is the side chain is already built into the starting material. You can make the alpha bromo compound and then displace that under SN2 conditions to get your alpha amino acid. And then the sequence as drawn would be racemic. So you would end up with both R and S at the alpha position. So just to sort of point out, again, you would just go to a chemical supplier and buy these things in the enantiopure form. But say you wanted to make, say you wanted fluorine, you wanted perfluorinated phenylalanine. You know, this would be a way to, you know, have that starting material make that for you. So those are typically not commercially available. And you wanted to study I'm running out of room here, but the drawing palette is blocking my way, so you can draw the other fluorines in there. If you wanted to see the effect of, okay, what's a fluorinated ring going to be on the properties of this peptide that I'm making, this could be a way to do that. Probably the easiest thing to do, because it gives you control of the what your side chain is. See the H is the Gabriel malonic ester synthesis. So the starting material on the left, so this is commercially available. And what you would do is, since that's an acidic proton, the pK is about 10, you would deprotonate that and then incorporate your side chain R using an electrophile with under SN2 conditions. And then from that, you would deprotect the imide And then your malonate here, we learned about hydrolysis and then decarboxylation. And so the product of that would be your amino acid. So that gives you a lot more flexibility in terms of what R is. So the last one is called the Strecker synthesis. This one incorporates the side chain R from the start, but the benefit of this is this reaction has been studied a lot. That when you do the condensation to form the imine and then you add cyanide to it, you can get enantiopure alpha aminonitriles. And then we know that the cyano group is a surrogate for an acid where you can hydrolyze it and still maintain the oxidation state of 3. So with those, you're basically, you have to have an aldehyde with your side chain in it. And so that would be just searching to see is that commercially available or not. So ultimately, practically speaking, those are ways that you would use to make unique or unnatural amino acids if a situation called for it. Any questions? So now, chemically speaking, if you're in the lab and you had to make a short sequence of amino acids to make a chain, how would we do that? So I'll give you conceptually how it's done, and then nowadays it's done with liquid handling robots, so it's all automated. Really, you just have cartridges with your amino acids. You plug the sequence in and it'll sort of do it for you. But this is how it does it. So if we want to make this sequence of AV, but just imagine it's a longer sequence, the concept still holds. This is the bond we're interested in making right there, the peptide bond. So that's a condensation. So if we split that, we're ending up with the alanine and the valine. So when you condense that alpha amino acid, so the amine with the carboxyl residue, it's a condensation in the sense that we're losing H2O. And so you'll see there's a lot of different peptide coupling reagents, and really what they're doing is a dehydration to form the peptide bond. But now the issue is, if you look at the starting materials, you have amine, amine, carboxyl, carboxyl. So really you can end up with 4 different products just using these things as drawn. This could couple with itself. That could couple with itself. this coupling with that to give us our desired product, or that coupling with that giving us a constitutional isomer. So to narrow down the possibilities, what we really have to do is we have to protect that amine so it's not nucleophilic, and then we have to protect that so it can't react with an amine to form a peptide bond. So really, outside of forming the bond, it's a protecting group strategy. So we'll talk about how they're coupled first and then the protecting groups necessary. So I'm going to do the cheap trick here of lasso chemistry. So there's your NH and your OH. So from those, we want to lasso out water and form that peptide bond. To do that, we've protected this as an ester, and we've protected that nitrogen as a carbamate. So that limits the amine on the valine and the acid on the alanine. Those are the only things that can now couple. And the reagent that will do that is called dicyclohexyl carbodiimide, abbreviated DCC. So notice the carbon here is sp hybridized, and it's at an oxidation state of 4. So it's an electrophilic carbon that basically wants to change. And so under the conditions, the mechanism for us isn't terribly important, but the oxygen of the acid will attack that to activate it. And then the nitrogen of the alpha amino acid is nucleophilic, which will then join those two amino acids together. So we're in the presence of a base, a very polar aprotic solvent at room temperature. And so if you look at the product, you know, the nitrogen's protected, and then the acid residue is also protected. And so to get to what we want, we just do 2 additional steps to remove those protecting groups. So I thought I'd spend 30 seconds or a minute talking about DCC. So again, these DCC doesn't exist in nature. You know, you can't go to a plant and extract it or anything. So it was developed by Professor John C. Sheehan at MIT, really for the aim of making penicillin in 1957. So nowadays, I mean, you can just go to Moffitt. If you're sick, they can give you penicillin. But back in the day, you know, it had to be, you know, harvested from mold or whatever it comes from. So people were interested in making it synthetically and then making derivatives of it that were more potent. And so part of the issue in making it was a coupling like this, a dehydrative coupling. And then Sheehan figured out that, you know, this fragment here of it, that's the carbodiimide part. That was crucial to getting this coupling to happen. The other part that I'll highlight in blue, nowadays that can be pretty much anything. Cyclohexane is nice because it's a lot of sp3 hybridized carbons, it's organic soluble, that sort of thing. You can change those groups around to make them water soluble, so that byproduct goes into the aqueous phase. You don't have to worry about purging it or extracting it. So that's the coupling. And now we need to talk about differentiating the amine and the carboxyl residues through protection. So a common protecting group is called BOC. So BOC anhydride. That structure is shown to the left here. It's abbreviated BOC2O. And essentially what it's doing is putting on, as I'll highlight in blue, the nitrogen's going to get that piece in blue. And since you're forming, it sort of looks like an amide, but overall the functional group itself is called a carbamate. The lone pair on nitrogen, due to that carbonyl, is not nucleophilic now due to resonance. So you completely take it out of play. And these reagents are really specific for amines to form these carbamates. And so that frees up your acid to do the coupling on that end. Other common nitrogen protecting groups: CBZ chloride and FMOC chloride. So these use a mechanism which we talked about already. That chlorine is a good leaving group. So the mechanism is nucleophilic acyl substitution. So the amine attacks, ultimately chlorine is ejected, and then you form these carbamates. And so the question would be, why would we need different nitrogen protecting groups? Ultimately, if you put the protecting group on, you have to remove it. And so all of these act under different conditions to remove them. The BOC group is removed with acidic conditions, so trifluoroacetic acid. So it says that the mechanism, you form that gas, you form CO2 gas, and then you end up with your ammonium salt. So for a cheap way of demonstrating this, so you can imagine the nitrogen being protonated. And then a base would abstract this proton to do an elimination. And so that's how you'd form isobutylene and CO2 gas. So we could say that trifluoroacetic acid is fairly acidic, but what if you don't want to use acidic conditions to deprotonate your sequence? I'll get to that in a minute, so we'll pause that question. Flipping over to the other side, the ester is just hydrolyzed. We called this saponification under basic conditions. So lithium hydroxide, THF water at room temperature, that will deprotect an ester. So that's pretty straightforward. So regarding the amine carbamate, so carbamate deprotection, so we use these abbreviations, NBOC, NCBZ, and FMOC. N just means nitrogen. So there's a menu here, acidic, neutral, or basic conditions. What is the rest of your molecule going to stand up to? So BOC is removed under acidic conditions with trifluoroacetic acid. The CBZ is removed by hydrogenolysis with H2 gas. And then FMOC is basic conditions with piperidine. So that's a nitrogen base, 6-membered nitrogen base. And so really you tailor your protecting groups ultimately to the sequence you're deploying. So if we consider two protected amino acids, N-BOC glycine and phenylalanine OME, that means this is a methyl ester. Which of the statements best describes the two functional groups that will react to form the dipeptide? So this is N-BOC glycine. And this is Phe-OME. So we see that the phenylalanine has the amine free, and then the N-BOC glycine has the acid free. So the sequence we would get then is N-BOC gly, Phe-OME. So that would say that A is the correct answer. So free acid reacting with free amine. And so you can see by picking out what we're protecting, that's going to then dictate what peptide bond is formed. Any questions? So conceptually, this is the strategy that you would use for solution phase. This just means like you're in the 256 lab and you got the chemicals and you're running them. So as opposed to what I'll talk about over the next couple slides, which is called solid phase. So really what we're doing in solution phase is you're starting from the N-terminus residue. So you start from the left, you protect that, and you're building the sequence from the left to the right. So when you protect that, that protects that amine, that frees up the acid portion of that. You're then going to couple on the next residue through the DCC coupling. And then you have to hydrolyze the ester of that dipeptide so that you can add in the next amino acid in the sequence. So really what you're doing is hydrolyzing, forming the peptide bond, and then repeating that sequence till you get to your desired chain length. And then ultimately you deprotect the N terminus. There's other things that can happen in here that are sort of beyond the scope of this course. But sometimes, you know, if you're starting with enantiopure amino acids, they can racemize, and that leads you to byproducts that you don't want. So you can imagine, you know, say a standard peptide coupling takes a couple hours. You know, if you were doing this for a living, you could maybe get 2 or 3 done per day. And so your chain would grow by 2 or 3 amino acids per day after you considered the reaction times, the workups, all of that. But if you're interested in making synthetic peptides, which are, you know, much longer than that, this would take forever. And so that was a motivating factor for changing the way this is done. And Merrifield won a Nobel Prize in 1984 for work that he had done, you know, in the 60s and 70s to basically automate this. And this is called solid phase peptide synthesis, or SPPS. And so solid phase, it's conceptually done different because you're starting at now the C terminus and you're working your way from the right to the left. And the reason for that is you're anchoring the C terminus onto a polystyrene bead, which is the solid phase. The advantages of this, like I said, nowadays there's machines you can buy that will do this for you, and you're just putting the liquid and different amino acids in there as different cartridges, which dispenses it automatically. And so it cuts down on the time requirement, and you can build up chains pretty quick. So how does this work? So as shown on the left here is 1-chloromethyl-4-vinyl benzene. So styrene itself has this structure. And really the alkene is used for polymerization. So the chloromethyl group, that's used as the anchor for your C terminus. So I show the polymer support, that repeating chain, as the bead. And then coming off each of those little beads is that anchor. And so you can anchor your C terminus to that, and then you're just growing the chain out. So the way that you get your C terminus on there is through an SN2 reaction of the carboxylate. So that's just the carboxylate displacing that chloride. Now it's anchored. And I'm showing this as like one, but imagine that bead is just surrounded by that first amino acid at the C terminus. And then if you've ever had like a white styrofoam cup, if you look at it close, you sort of see little circles. Like, consider that like a bead. And so off of each of those beads, you know, you have like millions of these anchor points. So if that's your R1 residue, now you're, again, building from the right to the left in terms of adding the amino acids. So you can see that the C terminus is anchored, so it's not reactive. Now this, you need to remove So remove the Bach group. That frees up the amine to do your next peptide coupling. So you have your beads. Essentially what's going to happen is it adds trifluoroacetic acid in there. It has an aging period to remove the Bach group. Now you have your free amine. And then you can add your next residue R2 through peptide coupling. So whatever your R2 group is, the DCC is added, the base, the solvent, that's now put on there. Notice this is also Bach protected. So at this point, you're just repeating that process. So you remove the Bach, couple on R3, remove that Bach, couple on R4, and then you grow your chain out. So ultimately, you have to get your sequence separated from the solid support. And so hydrofluoric acid is used for that. Essentially, you could think of it as that getting protonated. And then the F- anion cleaving it from the solid support. So this was demonstrated by Merrifield using or preparing a protein called ribonuclease. So that was published in the Journal of the American Chemical Society. And so ribonuclease is an enzyme that catalyzes degradation of RNA into smaller components. So in prior semesters, what I had done when I was using iClicker, this was like a bonus with free responses. I think I'll make it a bonus for your class, but I got to rejigger the, since it's not free response, I got to come up with the multiple choices. So I'll work over that over the holiday, and then I'll open that as a bonus. Because I really want you to read that paper to understand how this was done and the efficiencies that were gained, but also some of the issues. Because even though he did the method and he made that enzyme, he still had to prove that the synthetic enzyme was equally as active as the natural one. And so it's an interesting paper to read, and I think knowing what you know, you should be able to understand everything that was done, which again is, it's a paper that provided evidence for a Nobel Prize. So don't think that the science of a Nobel Prize is beyond your understanding. Like, you know enough now to understand the logic by which he was operating. And the motivation was, we need to do this quicker. And so it was just, really a nucleation point of some basic science, just automating it. Any questions? I know we're chugging through here. We could definitely take more time on this stuff, but again, since it's not on an exam, really it's just to give you a broad overview. So this was a YouTube video. I can't play it because I'm in a PDF here, but I think these are linked into Canvas. And I would definitely encourage you to take some time watching these. They're not that long. But they use some nice animation to describe what are the elements of a protein and what do proteins do. And so now that you know how to make these sequences, ultimately, how are they impactful for proteins and enzymes? So we'll get over the next, what do we have, 24 minutes here, something like that. Just a brief overview of how the sequences matter and ultimately why these enzymes catalyze reactions in the first place. What do they do? So to understand now what's happening from like the small organic perspective to the large biochem perspective, we really need to drill down into the amide functional group and how that will ultimately impact secondary and tertiary structure. So why are proteins even made of amides to begin with? We saw in Module 3 that the amide functional group was the least reactive towards nucleophilic acyl substitution. So again, there's oxidation state 3 of the amide. And we saw that if you're starting with an amide, it's really hard to substitute that for another nucleophile. The reason being, from the organic perspective, is that the byproduct is an amide base. And the pK of that conjugate acid is about 38. So that base is really strong, meaning that the acid is pretty weak. So it's not a good leaving group. So we can infer from that this is a very strong bond. And again, whether you believe in religion or creation or evolution, these are the things that hold us together, that strong bond. Like, we're not made of like acid chlorides because we would just fall apart. So these sequences of these peptide bonds are there because that's the strongest bond of oxidation state 3. And so some organic 1 stuff, if you were to cleave this bond homolytically, that would require 400 kilojoules per energy of to break that, where each atom, the C and the N, both get an electron. So it's a pretty strong bond. The other factor of the amide bond is resonance. And this really impacts, again, now the secondary and tertiary structure. So we know that nitrogen has a lone pair. We know that CNOF, that increases electronegativity. So oxygen's more electronegative. So amides, of all the oxidation state 3 functional groups, the resonance form really matters, where this now looks more like a pi bond, and that looks more like a sigma bond. And so remember, that always helped us explain why the amide carbonyls are always less than 1700 in the IR, because they're more sigma bond-like now. As a fallout of that, you know, when you look at this resonance form, you say that's a sigma bond. You know, it should rotate, meaning that those two methyl groups are the same. But in the resonance form, now they're different because that oxygen is close, or that methyl group is closer to that oxygen. And then this methyl group is opposite. So in the NMR spectrum of DMF, you actually see 3 peaks rather than 2 at 6 to 1. So you essentially see 3 singlets because of their relationship relative to the oxygen of the carbonyl. So now that's important, even though I'm showing DMF, it's important in your peptide chain because all of those are peptide bonds. And then that's going to matter what side of the carbonyl the side chain is lying. So your first approximation, you'd expect to see 2, but in reality, you see 3 due to resonance. And again, that is due to, this being more electronegative than N. So oxygen being more electronegative than nitrogen is going to push that lone pair up onto the oxygen. And so here is where it ultimately falls out. So in a dipeptide, notice the disposition of the side chains here. On the left structure, when everything is sort of zigzag, you have R1 and R2 like on opposite faces of that plane. If you were to rotate around the sigma bond, now R1 and R2 are in the same plane, and that's a steric clash. If you think of those two structures as having the double bond between the N and the C, then you can assign cis and trans. And so the trans is favored over the cis by about 8 kilojoules per mole. Because when they're on the same, when R1 and R2 are on the same plane, that's when they start banging into each other. And so to a point, you can imagine, as the chain grows out, the chain is eventually going to get floppy, right? Eventually, not necessarily through like a cis geometry, but it's going to start curving back around. And then when it starts doing that, now you get secondary interactions. And the secondary interactions are ultimately what is going to give the protein its shape. And then shape will determine its function. So I cracked open this book. I don't know what they use in the biochem sequence here, but this is the book that I have. It's pretty common, Voigt and Voigt. What you see in those olive-colored rectangles are the peptide bonds that are showing the trans geometry. So because they're all trans, you just get that straight chain. It doesn't kink back around at all. And then the hollow colored atoms, those are the side chains pointing out. So you can see that as you go along, they alternate from the top or the bottom on that chain. So in zooming into a dipeptide part of that, there are two main angles that help determine shape. So rotation about the carbonyl to the alpha carbon, and then the alpha carbon to the nitrogen. So depending on the way these things rotate, again, that's going to help determine the ultimate shape. And hopefully I can remember this name right. It's called a Ramachandran plot, where basically you plot these different angles, and then what you'll see is that for the amino acids involved, there's only certain regions of this plot in which you'll see points. And those points, again, are indicative of what sort of shape can happen at that minimum. And so ultimately, it's about minimizing any... Are you texting without looking at your phone? That's amazing. It's like...
Yeah, I can do it on like my actual laptop too.
That's pretty wild.
I mean, occasionally I'll make mistakes, but that's what we have spell check for.
Yeah, exactly. So as the peptide chain grows longer and longer, it gets floppy, right? Regardless of the trans configuration. So the floppiness impacts the secondary structure, as we'll see, due to the non-covalent interactions that will form. And so these are just some Newman projections of what we're looking at for those two different angles. What are we trying to minimize? So from the alpha carbon down to the amine group of the amide, so the nitrogen is in back here, you're looking at how that next amino acid is going to sit relative to that carbonyl. And then the same thing from the alpha carbon down to the amide carbonyl. How is this rotation going to play out to minimize steric interactions? And again, when this rotation happens, again, there's going to be energy local minima and maxima. So it's only going to sit sort of in a certain dihedral angle that's favorable to the functional groups present. So as we've seen, the primary structure is the exact sequence of amino acids. The secondary structure is the localized 3D structure produced by the conformations and non-covalent interactions. So those are called NCIs. So two main things you'll want to be familiar with are what are called alpha helices and then beta pleated sheets. So the sequence is, made up of the amino acids, but then they start forming these other sort of structures that result from non-covalent interactions. The tertiary structure is the overall three-dimensional shape, and then quaternary is the association of two or more polypeptide chains in a protein complex. So what are the interactions? Covalent, that's your typical covalent bond energies from 150 to 500. And then you see the next four, those are non-covalent interactions. So there's salt bridge, that's, you know, like a carboxyl and an ammonium, so something like this. A hydrogen bond, we've seen that throughout organic 1 and 2. Hydrophobic interactions, excluding water. Van der Waals interaction, that's these temporary, you know, fleeting interactions that exist between polarized bonds. So just temporarily induced, like Coulombic interactions. Now, even though most of these non-covalent interactions have weak bond energies, you know, if you think of a protein being hundreds or thousands of amino acids, you get a lot of these small numbers adding up to a large overall energy interaction. So visually, what do these things look like? So a beta pleated sheet has linear sequences running. There's two ways to describe this. Even though the chains run parallel, they can be described as parallel or non-parallel based upon the N to C, C to N, how you're looking at that. So really you have these parallel lines of the amino acids that form a sheet. And so this dotted line here, those are hydrogen bonds. So basically you have NH hydrogen bonding to a carbonyl on the other strand. Alternatively, the alpha helix, so that's, you know, if you think of DNA as a helix, or if you think of like your standard sort of screw that has threads. So that spiral, that's the helix. So you have these turns, and then they hydrogen bond from sort of one step down to the next, or bottom to top. So that's what's indicated here. So there's beta sheets, alpha helices, and as you see in the ribbon, so that's typically how proteins will be designated, with these ribbon structures. Like this, for example, here would be a turn. Then you see the beta pleated sheet, alpha helix. We talked about disulfide bridges, other types of hydrogen bonding. Hydrophobic interactions, that's when you have like non-polar amino acids pointed sort of out. Hydrophobic interactions and salt bridges. So on the tertiary structure, there's a salt bridge right there. So you have your carboxylate ammonium attraction. So that's like an ionic attraction. And again, what you notice from that, so see that turn? Like normally if these things are far apart, but then they're wiggling around and they find each other, now that straight chain is formed to turn into that larger peptide structure. So since we're building up towards ultimately looking at like DNA, I thought it would be nice to include a couple slides on helical chirality. So if you've taken physics, you probably learned about like the right-hand curl rule. So if you take your hands and curl them, and then you look at your thumbs, like those are enantiomers. Anyone do any sort of carpentry? So a normal like threaded screw you buy from the Home Depot or Lowe's, if you want, what's the rule? If you want to drive the screw into the wood, righty tighty, lefty loosey. So if you're taking it, you're turning your hand, what is that? Counterclockwise, right? So a question I always ask students is, you could have an enantiomeric screw, right? But then you'd have to use your left hand to drive it in. But most of the screws that are sold are right-handed. Why do you think that is? Most people are right-handed. And the other thing is that direction, if you think about it, when you're turning it, you engage that muscle the most, right? That's where the meat of your forearm is. And so that gives you the push in. And so if you look at this, that helix, that's a right-handed helix. Because if you were to put your hand here, you put your hand here and turn it that way. If you imagine that's the point, that's going to drive it into the wood. So, you know, you learned about point chirality, which is sp3 hybridized carbon. Helical chirality is not point. So it's a little harder to understand in terms of, if I just gave you two helices, figure out what the direction is. It takes a lot more challenging. I don't have a picture of it, but my daughter has curly hair, and she has this one curl that comes off her like forehead, and I was thinking, what is the direction of that coil of that, you know, that curl? And I think it was a left-handed one, just based on looking at it. But it does take some effort to figure out helical chirality. So again, there's right and left-handed helices. And so they're described by the number of peptide units per turn. And then the rise is the distance per turn. So that means, like your starting point, say like that little thing there, what is the distance going up to where you see that next turn starting? So that would be the P. And then how many amino acids are in that full turn? And then that will translate to when we start talking about DNA base pairs as well. So as it says, the right-handed alpha helix is the most commonly observed with L amino acids. And then I think DNA is also a right-handed helix as well. So that then begs the question, not that we'll answer it in this class, but like, you know, how did it get that way from, you know, this abiogenesis question? Whether you believe in that or creation. There for today, I think we did pretty good, even though it was fast. But ultimately, again, you know, we're looking at how can we make these things into proteins, and then coming back after Thanksgiving break, we'll look at, okay, now how does nature make these proteins from reading DNA? And then that will be two of your other activities. I think we'll stop.